Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the topic is related to microbiology, and the title of my today's lecture is citrate test and its mechanism. Basically, it's a biochemical test and it is used to differentiate between microorganisms and those microorganisms that are citrate positive. They consist of a special type of enzyme that is known as citrate permease, and they can only utilize citrate as their sole carbon. And a microorganism that cannot utilize this citrate, so they lack that enzyme. So just because to detect that enzyme, we use this biochemical test. Okay. So in today's lecture, I will discuss the different steps that are involved in the preparation of the citrate media, and I will also discuss the basic principle of it, that why we are conducting it, and at the last I will show you the different chemical reactions that are mainly taking place inside the tubes. Okay, and that part is a very much important for microbiology students, because knowing about the chemical reaction is very much important. Okay, so let's start with the video lecture. First of all, you should know about the principle, as I have told you that citrate agar is used to test an organism ability to utilize citrate as a source of energy. Okay, so these microorganisms can only utilize citrate as their source of energy, as their source of carbon. Okay, so when we are applying the media, when we are giving the media, so that media consists only of citrate. No, there is no glucose, there is no sucrose and different other glucose. Only citrate is present. Okay, the medium contains citrate as a sole carbon, as I have told you, and different ammonium salts are also there that are used as a source of nitrogen, because nitrogen is used to produce and to manufacture different nitrogenous compounds like amino acids and nitrogenous bases. Okay, so citrate is used as a sole carbon, and ammonium salts are used as a source of nitrogen. Okay, this test is among a suite of IMVs, IC tests. Okay, this stand for indole, methyl red, Voges prosper, and citrate. So these tests, when you heard this such type of terms, so you can easily tell that these are four types of tests, and citrate is also included in these tests. Okay, it can come in the MCQs, and it is used to differentiate among the gram-negative bacteria that is related to the family Enterobacteriaceae. Okay, so this citrate test is used to differentiate between different gram-negative bacteria and the members of Enterobacteriaceae. First of all, you should know about the composition of it. When you see a bottle of citrate media, or basically there is semen citrate agar is the name is used, placed on the bottle. Okay, so what is the composition of it? So it consists of sodium chloride that provide different salts, sodium and chloride, to the microorganism. Sodium citrate is there, as I have told you that it is used as a sole carbon source. Ammonium dihydrogen phosphate, it is used as a source of nitrogen, and dipotassium phosphate and magnesium sulfate are used as a cofactor for the enzymes. Okay, the potassium and the magnesium are used as a cofactor for the enzymes to active to perform its activity. Okay, and bromotyl blue, it is used as an indicator. Okay, it is used as an pH indicator. So when there is a certain shift in the pH, so it will change its color, and by detecting that color, we can easily tell that either the organism is citrate positive or citrate negative. Okay, and at the last, it consists of agar, just to it performs solidification of the media. Okay, so it also consists of agar. If I show you the preparation of animated form, so first you have to take the powder media, and you have to dissolve it in the distilled water. After that, you have to cover it and place it in an autoclave. Okay, just to remove all the microorganism from it. So you have to sterilize the media. You should also sterilize the test tubes. Okay, because we have to pour the media into the test tube. So just look here to this technician, this microbiologist. He is pouring the media into the test tubes. Okay, so after that you have to then incubate it. Okay, you have to place the, sorry, the color of the media is just like this. Okay, if it is it is just like dark green in color. After that, what will you do? You have to streak a specific colony into the test tubes. So you have to take a unknown colony from the petri plate from by using a inoculating loop, and then you have to dip it into the citrate media. After that, but keep this thing in mind that your inoculating loop should not touch the end of the bottom of the test tube. After inoculating it, you have to streak the inoculating loop over the slant. Okay, this curve type media is known as slant, and this is performed after the when the media is solidified. Okay, so keep this thing in mind. You should not apply it when it is still in liquid form. When it is solidified, the temperature get lower, so after that you have to inoculate the media, and then you have to put it in the incubator for 1 day, 2 day, or 3 day, but mostly 3 days are important for it. So you have to place it these test tubes in the incubator for 3 days. Now here after 3 days you can see different results. Okay, so if your test tube is pure green in color as it was before before the inoculation, so your test will be negative. Okay, so the bacteria that you have used in the test tube is negative, and if it has transformed, if it changes its color to blue, so then you can say that my bacteria is citrate positive. So the main thing is that why this blue color appears, and why in the negative citrate test tube, why there is no color production, and in the positive, why there is blue color production. Okay, so the main aim of this video is to understand the results. Okay, so just look here, this is the preparation in the text form. You can take a screenshot and then you can study it. As I have told you in the animated form, it is just all like that. Okay, so these all are the text form. Now what basically are the chemical reactions and why these colors appear? Okay, so keep this thing in mind that when, for example, when your bacteria consists of a citrate enzyme. Okay, because we are detecting the citrate enzyme. So if your bacteria consists of a citrate enzyme, so what will it do? It will take the citrate that is present in the media and will by the action of citrate permease, it will the citrate will go inside the bacteria. After that, by the action of citrate enzyme, what will happen? It will metabolize those citrate. Okay, first it will be converted into oxaloacetic acid, and also for byproduct acetic acid will be produced. After that, what will happen? Just look here to this portion. Then it will be converted into pyruvic acid. Okay, then it will be converted into pyruvic acid, and carbon dioxide will also be produced. Okay, so up till here, just keep this thing in mind. When there is citrase, okay, this is our main enzyme. If it is present in the microorganism, so what will happen? It can break down the citric acid to oxaloacetate, and after that the oxaloacetate can be converted into pyruvic acid and carbon dioxide. Okay, so don't be confused by these chemical reaction. Okay, just focus on the pyruvic acid. uh, After that, what will happen if you're having a pyruvic acid? Okay, wait a minute. Okay, so what is the biochemistry within the bacteria? Okay, what is basically the biochemistry within the bacteria? So what will happen, that sodium citrate, basically we are taking the citrate that is conjugated with the sodium. Okay, Na. So what will happen, that the sodium citrate will be converted into pyruvic acid, and it will release its Na. Okay, it's sodium. Okay, it will release its sodium, and also carbon dioxide is produced and oxaloacetate is produced, as I have shown you in the molecular structures. So what will happen? that the sodium that is released from the citric acid will then react with the carbon dioxide and water that is present in the media. Okay, it will the carbon dioxide is basically this one. Okay, this one is the carbon dioxide. Okay, so it will react with the carbon dioxide. Okay, this Na will react with the carbon dioxide, and it will produce these such type of Na2CO3 alkaline products. Okay, so this is an alkaline product, so the media will become alkaline. Okay, so the pH will increase. Another thing is that the ammonium salts that were used as a nitrogenous source, also they will be metabolized, okay, in order to give nitrogen to the microorganism and to manufacture different nitrogenous compounds like amino acids and nitrogenous bases. Okay, so they will also use the nitrogen as the nitrogen source, when different byproducts will also form that will be having a basic nature. Okay, so this Na2CO3 sodium bicarbonate, sodium carbonate is also released, and also other byproducts that are alkaline in nature is also released. So the pH increases, and when the pH increases, so the bromomethyl, that the indicator, what will happen that it will change its color, and it will change the color from green to blue. Okay, so just because of the production of alkaline products, the color changes from green to blue. So we can easily tell that our bacteria is citrate positive. Okay, and this was all just because of the production of pyruvic acid from the citrate, and this was all just because of the citrase enzyme. Okay, so if there was no citrate enzyme, okay, if there was no citrate enzyme, so what will happen? There will be, if there was no citrate enzyme, so there will be no production of pyruvic acid, and there will be no carbon dioxide, there will be no sodium. Okay, there will be no carbon dioxide, so there will be no formation of this sodium carbonate, as well as the ammonium salt that are used as a nitrogen compound, nitrogen sources, will also not break down. Okay, because one thing, if you know that pyruvic acid is mainly performing as a precursor metabolite for the production of different amino acids and different nitrogenous bases. Okay, well, this pyruvic acid is converted into acetyl CoA, as you have remembered in the Krebs cycle. So acetyl CoA, I'm writing just A and CoA. Okay, acetyl CoA. So it is converted into acetyl CoA, and after that, this acetyl CoA reacts with different nitrogenous compounds to produce different amino acids and different nitrogenous bases. Okay, so if there is no pyruvic acid, there will be no acetyl CoA, and these ammonium salt cannot be broken down and cannot react with the acetyl CoA, and it will not form any byproduct. Okay, this is very much technical condition of this citrate test. Okay, so there will be no nitrogenous compounds and no sodium carbonate, so there will be no increase in the pH, and the and the color of the media will still remain green. Okay, so that's why you have to look to the both sides. If it is positive, so why it is positive? If it is negative, so why it is negative? So this was all because of this enzyme. Okay, if it is not present, so there will be no pyruvic acid. This pyruvic acid will not be converted into acetyl CoA, and the ammonium salt cannot react with the acetyl CoA because there is no acetyl CoA, and it cannot it cannot produce different nitrogenous compounds. Okay, because there is no precursor metabolite. So if there is no precursor metabolite, so to whom will they react? Okay, so that's why no byproduct will form, and if there is no citrate enzyme, so sodium citrate cannot be broken down, and there will be no excess of sodium. Okay, there will be no excess of sodium, and there will be no carbon dioxide, and there will be no sodium carbonate. Okay, so the color of the media will remain green in color. Okay, so keep this thing in mind. Now here come that these are the microorganisms that are showing positive test results for the citrate test, and these are the microorganisms that are showing negative test results for the citrate test. Okay, so this was all about the citrate test and its mechanism. Hope you have understand this video well. If you have any questions, so you can ask me in the comment box, and if you have understand it, so just like this video, share it with your friend, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.